My name is James Watts Compton, science researcher and book author. When talking about the origin of life, many believe that the issue is still a scientifically open question, that science is working towards solving the problem and a solution near the next corner. Lee Cronin, for example, made a prediction in 2011.
Just a quick question on timeline. I mean, you believe you're going to be successful in this project? When?
So many people think that life took millions of years to kick in. We're proposing to do it in just a few hours, once we've set up the right chemistry.
And when do you think that will happen?
Hopefully within the next 2 years.
That would be a big story.
Unfortunately, it didn't concretize. There are many reasons why intelligent guidance had to be involved in the process. In this video, I will expose one reason. One of the most popular hypotheses is that life started with the so-called RNA world. Before we continue, a quick elucidation of what life is. As the shortest description, life is basically chemistry and information. More extendedly, life consists of reproduction, metabolism, nutrition, complexity, organization, growth and development, storing and transmitting instructional complex information, hardware, software entanglement, permanence, and change. Donald Prothero wrote in his book, The Evolving Earth, published in 2019, the simplest scenario for the origin of living, self-replicating systems would be an RNA world. The very first self-replicating form of life would be a single-stranded RNA, perhaps enclosed in a lipid bilayer membrane and perhaps using simple carbohydrates for food storage. Using both its replication powers and enzymatic powers, it would make more copies of itself and perform the role of the proteins as well until later, more complex reactions involving many different proteins could evolve. Every year, more discoveries are made that add details to our understanding of the origin of life and the RNA world. He starts his narrative with a single-stranded RNA. Here's the problem. In order for the RNA world to be true, one needs to start much earlier, namely with the origin of RNA itself. Only then, further questions can be asked, like how RNA polymerized and eventually started self-replicating. A number of reasons have been given why a prebiotic synthesis of RNA, and even more, DNA, is too complex. In cells, the synthesis of RNA and DNA requires extremely complex energy-demanding, finely adjusted, monitored, and controlled anabolic pathways. Since they were not extant prebiotically, RNA had to be synthesized spontaneously on early Earth by abiotic alternative non-enzymatic pathways. This is one of the major, among many other unsolved origin of life problems. A recent article in the Journal of the Royal Society claims that RNA, proteins, and the genetic code that binds them each look like products of natural selection. I have no idea how they conclude that they seem the product of natural selection. To me, it indeed seems that these building blocks had to be selected, but nature has no intent, no goals, no aim to complexify molecules towards life. If a selection process was necessary, then it had to have another source, instead of nature. What is missing from this story of the evolution of life on Earth is the original means of producing such sophisticated materials as RNA. The main problem is that the replication of RNA depends on a clean supply of rather complicated monomers, activated nucleotides. What was required to set the scene for an RNA world was a highly competent, long-term means of production of nucleotides. In practice, the discrimination required to make nucleotide parts cleanly, or to assemble them correctly, seems insufficient.
Hi. I am Graham Ken Smith. Organic chemist, molecular biologist, and author of several books on the origin of life. So powerful has been the effect of Miller's experiment on the scientific imagination that to read some of the literature on the origin of life, including many elementary texts, you might think that it had been well demonstrated that nucleotides were probable constituents of a primordial soup, and hence that prevital nucleic acid replication was a plausible speculation based on the results of experiments. There have indeed been many interesting and detailed experiments in this area. But the importance of this work lies, to my mind, not in demonstrating how nucleotides could have formed on the primitive Earth, but in precisely the opposite. These experiments allow us to see, in much greater detail than would otherwise have been possible, just why prevital nucleic acids are highly implausible. Let us consider some of the difficulties to make RNA and DNA. In my book, genetic takeover. I listed 14 major hurdles that would have to be overcome for prime nucleotides to have been made on the primitive Earth, from the buildup of sufficient and separate concentrations of formaldehyde and cyanide to the final winding up of the nucleotides. In practice, each of these processes would be subdivided into separate unit operations that would have to be suitably sequenced. I think, to say that on average the 14 hurdles that I referred to in the making of prime nucleotides would each take 10 unit operations, that at least 140 little events would have to be appropriately sequenced. If you doubt this, go and watch an organic chemist at work. Look at all the things he actually does in bringing about what he would describe as one step in an organic synthesis. This is a huge number, represented approximately by a 1 followed by 109 zeros. This is the sort of number of trials that you would have to make to have a reasonable chance of hitting on the one outcome that represents success. Throwing one dice once a second for the period of the Earth's history would only let you get through about 10 to the power of 15 trials.
An article published in 2014 summarizes the current status quo. The first, and in some ways, the most important, problem facing the RNA world is the difficulty of prebiotic synthesis of RNA. This point has remained a focal point of the efforts of prebiotic chemists for decades. The traditional thinking was that if one could assemble a ribose sugar, a nucleobase, and a phosphate, then a nucleotide could arise through the creation of a glycosidic bond and a phosphodiester bond. If nucleotides were then chemically activated in some form, then they could polymerize into an RNA chain. Each of these synthetic events poses tremendous hurdles for the prebiotic Earth, not to mention the often invoked critique of the inherent instability of RNA in an aqueous solution. Thus, the issue arises of whether there could have been a single environment in which all these steps took place. Benner has eloquently noted that single-pot reactions of sufficient complexity lead to asphaltization, basically, the production of intractable goo. The asphalt problem has been outlined by Rob Stadler and Change Loratan in their book, Stairway to Life. Even in a very short DNA of just 2 nucleotides, there are dozens of incorrect possible arrangements of the components, and only one correct arrangement. The probability of consistent arrangement decreases exponentially as the DNA lengthens. If natural processes could polymerize these monomers, the result would be chaotic asphalt, not highly organized, perfectly consistent biopolymers. Think about it. If monomers spontaneously polymerized within cells, the cell would die because all monomers would be combined into useless random arrangements. And Steve Benner has made another observation, which means a death knell for the prebiotic synthesis or RNA. An enormous amount of empirical data has established, as a rule, that organic systems, given energy and left to themselves, devolve to give uselessly complex mixtures, asphalts. The literature reports, to our knowledge, exactly 0 confirmed observations where evolution emerged spontaneously from a devolving chemical system. Further, chemical theories, including the second law of thermodynamics, bonding theory that describes the space accessible to sets of atoms, and structure theory requiring that replication systems occupy only tiny fractions of that space, suggest that it is impossible for any non-living chemical system to escape devolution to enter into the Darwinian world of the living. We are now 60 years into the modern era of prebiotic chemistry. That era has produced tens of thousands of papers attempting to define processes by which molecules that look like biology might arise from molecules that do not look like biology. For the most part, these papers report success in the sense that those papers define the term. And yet, the problem remains unsolved. Another widespread myth is that RNA polymerization is something that has been demonstrated to be possible on clay. Robert Shapiro wrote a critique in regards to prebiotic proposals of clay-catalyzed oligonucleotide synthesis. An extensive series of studies on the polymerization of activated RNA monomers has been carried out by Ferris and his collaborators. A recent publication from this group concluded with the statement, the generation of RNAs with chain lengths greater than 40 oligomers would have been long enough to initiate the first life on Earth. Do natural clays catalyze this reaction? The attractiveness of this oligonucleotide synthesis rests in part on the ready availability of the catalyst. Montmorillonite is widely distributed in deposits on the contemporary Earth. If the polymerization of RNA subunits was a common property of this native mineral, the case for RNA at the start of life would be greatly enhanced. However, the catalytic activity of native montmorillonites is very poor. They interfere with phosphorylation reactions. The activity of the montmorillonite depended strongly on its physical source, with samples from Wyoming yielding the best results. Eventually, the experimenters settled on vol clay, a commercially processed Wyoming montmorillonite provided by the American Colloid Company. And now I will address the last claim, namely self-replication. The RNA world is a hypothetical stage in which self-replicating RNA molecules proliferated on the early Earth. The term RNA world was coined by American molecular biologist Walter Gilbert in 1986. The hope was that evidence would be found demonstrating the ability of RNA to self-replicate. This would be the initial state of self-replication in a hypothetical origin of life scenario. An article in 2017 stated, Gilbert himself expressed some disappointment that after 30 years, a self-replicating RNA has not yet been synthesized or discovered in the years since he predicted his hypothesis, but he remains optimistic that it will emerge eventually. Many found the metaphor appealing, a world with a jack-of-all-trades RNA molecule, catalyzing the formation of indispensable cellular scaffolds, from which somehow then cells emerged. Others were quick to notice several difficulties with that scenario. These included the lack of templates enabling the polymerization of RNA in the prebiotic complex mixture and RNA's extreme lability at moderate to high temperatures and susceptibility to base-catalyzed hydrolysis. To go from a self-replicating RNA molecule to a self-replicating cell, which is in a literal sense a chemical factory, depending on at least the information stored in 500,000 nucleotides, is a huge gap that has not been addressed. For a factory to make a duplicate, a copy of itself, it must employ and use a description of itself. This description, being a part of the original factory, must itself be prescribed by something else that is not itself. That is, it must come from the outside. Why? In order to describe something, one needs to be a conscious agent, able to do so. If the factory itself was not the conscious agent, being able to observe itself and describe itself, it must have been something else. I, as a human being, conscious as I am, can observe and describe myself. A non-conscious something has never been observed having these necessary cognitive and intellectual capabilities. That's why the origin of biological information stored in genes is a hard, untractable problem for naturalists. It cannot be answered unless one acknowledges an intelligent designer. The description of itself, in order to make a copy of itself, must be expressed. Parts, subunits, or an agglomeration of building blocks do not comprehend how they could shape themselves to move into the right functional form and position, become a subunit, and join to become part of a functional interlocked complex system. So in order to do so, and have the know-how to get energy directed precisely where needed to have the device performing and exercising its tasks, an organizing principle, and instructional assembly information to create such an irreducibly complex system must be there to direct the process and come from the outside. In the cell, this information is stored in genes. Unless that information directs the assembly and energy that moves the system away from equilibrium to perform its work, the individual parts would either lay around and remain unorderly, in a chaotic state, would remain non-assembled, disintegrate, or, eventually, driven by random external forces, join into a basically infinite number of non-functional chaotic aggregational states. Creating an interdependent system where the information, stored in hardware, is encoded, transmitted, and decoded to direct the assembly process of the identical representation of that digital information into an analog 3D form, the physical reality of that description adds untractable difficulty for unguided processes. Furthermore, in order to have a copy, a daughter cell, identical to the mother cell, the description of itself must be passed to the offspring. Only then, a replica can be completed. The cause leading to a machine's and factory's functionality and self-replication has only been found in the mind of intelligent engineers with foresight, intent, and goals, and nowhere else. Memory-stored controls transform symbols into physical states. Von Neumann made no suggestion as to how these symbolic and material functions in life could have originated. He felt
that they should occur in the world at all is a miracle of the first magnitude.
The origin of RNA is just a tiny problem compared to the huge number of things that have to be explained for life to start. If you want to know more, you can buy the book, On the Origin of Life and Virus World by Means of an Intelligent Designer, The Factory Maker, Paley's Watchmaker Argument 2.0, where not only the problem of the origin of RNA is addressed, but many other origin of life issues. Thanks for watching, and please subscribe.